DNA is the template from which RNA is made. Nucleotides form conjugate pairs. A goes to U, C goes to G, and vice versa. RNA polymerase binds at a specific location and orientation on the promoter. This determines where transcription starts and which strand of DNA will be the template strand. As we can see, this is the strand of RNA, and this is the template DNA strand. The mRNA moves through the nuclear envelope and into the ribosome. The mRNA must enter the ribosome 5' end first. Amino acyl tRNA synthase uses an enzyme and ATP, attaches an amino acid to its tRNA. On an mRNA strand, every group of 3 bases is a codon, and on a tRNA, every group of 3 bases is an anticodon. As you can see, the conjugate pairs of nucleotides match up. As you can see, the A site holds the next tRNA, carrying the correct amino acid to be bonded to the growing polypeptide chain. The P site holds the growing polypeptide chain connected to the correct tRNA. The E site is what holds the tRNA that has already given its amino acid to the growing polypeptide. In the elongation stage of translation, amino acids are added to the previous amino acid of the growing chain. This is the growing chain. And this is the next amino acid to be added. The ribosome is the location of protein synthesis. The mRNA always moves 5' end first through the ribosome. Keep in mind, peptide bonds are what hold the polypeptide together. These are the peptide bonds. 1, 2, 3. The final step of translation is termination. A stop codon, such as UAA, reaches the A site of the ribosome. It acts as a signal to stop translation. Once it gets scanned, a releasing factor, which is shaped like a protein, will bind directly to the stop codon in the A site. The release factor hydrolyzes the bond between the tRNA and the complete polypeptide, thus allowing the fully grown polypeptide to be released through the exit tunnel.